see students. Okay. My screen is visible, right?
Yes, Tina.
Okay, great. Who's this? I heard a new voice.
It's Shubha.
Okay, did I speak to you last class?
Yes.
Okay, great. Hi, Shubha.
Hi, Tina.
Okay. So, so you're also an integrated student, right?
Yes.
Okay, so today, so in our last class, we actually covered the basic, um, the basic body plan and the basic events that will, in morphogenetic movements, that will give rise to the transition the embryo into an adult fly. So starting from the egg laying, undergoing the different divisions, cellular divisions, which are quite distinct in Drosophila because it's just in its nuclear divisions as opposed to other systems where it's a proper cellular divisions, but here only the chromatin divisions happen. The nuclear material undergoes just S and M, and undergoes synthesis and mitosis, till later on when the actual cellular divisions kick in. And at the same time, you have the handing over of the maternal components. I mean, initially all the majority of the events are under maternal control. So the mom lays in RNA and proteins that support the first few hours of cellular division, first few hours of division and development in the embryo. Following stage 10 or 11, 13 and 14 is when actually the maternal to zygotic genome activation happens, transition happens, and the zygotic genome is now activated, takes over, the gastrulation events of gastrulation are initiated, and then gastrulation will give rise to all the different, give rise to the segmented body plan eventually. So first of all, it will delineate the 3 germ layers, and then the extended morphogenetic movements that take place will give rise to the segmented body plan of the animal. And the first insta, the embryonic development stops with that, and the embryo now more or less looks like a larvae, which is going to hatch as a first insta larvae. So, so these are the events that I just summarized. We spoke about last class. Today, we will cover how Drosophila as a model system proved to be very valuable in understanding how these processes are being generated. So in a way that, I mean, how are these things even happening? What is the molecular genetic basis of these events, and how are they being controlled? How is development of this animal happening, and what are the rules that the animal follows to be able to develop like the way it did? Now, much of the understanding...
Hello.
Yeah, go ahead.
Yeah, Tina, sorry to interrupt you, but last time you had left us with this question that why this development in Drosophila follows.
Yes. I will. Can I? Maybe we'll come to that in the end.
Yeah, sure. Okay.
Yeah, I thought I will just go over the class and then we can discuss on that at the end. I'm so glad you brought it up. So glad. So we'll do that in the end.
Okay. It's okay.
Yeah. Okay. Great. Okay. So who was this, by the way?
Atal. I'm in PhD batch from.
Yeah, what's your name?
Atal.
Atal. Okay. Thanks, Atal. Okay. So essentially in today's class that what we would like to understand is how is the animal developing through molecular means. And while visualization of these steps in embryogenesis were done much earlier, it is only in 1990s, like I mentioned last class, that Drosophila development, understanding Drosophila development was really bolstered by the advent of molecular biology, cloning, and techniques involved in molecular cloning, sequencing, not sequencing, but enhancers, identifying transcriptional tools that became available, actually led to the identification of genes. So what Drosophilists were doing back then is to randomly mutagenize flies. But what exactly was the gene product that got mutagenized was not clear. And with the advent of molecular cloning, these identifications became very valuable because you could directly pinpoint the mutation in a gene. You could name the gene and then say, oh, okay, this is the mutation in hedgehog, or this is the mutation in toad, or this is the mutation in dorsal, which results in this particular phenotype. So you could now be able to affirmatively contribute the phenotype arising from a defect in a gene product. And that then revolutionized the whole idea of development and how development was being controlled and what are the underlying principles. Now, and that led to the series of the Nobel Prize that was given to Ed Lewis, Muslim Follard, and Eric Wieschaus. And so this is like the yeah, was shared amongst all three of them. Okay, now so essentially, understanding Drosophila development would mean where do we start from? So you have the embryo, which is post-fertilization, and then the series of events that take place are seen. Now, are they determined by the embryonic components, or is it the component that the mother contributes to? So meaning, does the genotype of the mother affect what the embryo is going to be doing? I mean, that's the answer to that is yes, because initially the components in the embryo, before the zygotic genome activation happens, everything is decided, is or taken care of by the components that the mother lays in. So if the maternal so this is called the maternal component. Now, if the maternal components have any kind of defect, then you can imagine that the whole early embryonic development will be affected. So now, to what extent does the maternal component contribute to the embryonic development? So what do the RNA or the protein components that the mom deposits into the egg set up? So essentially, it does a very valuable job for the embryo because it sets up the coordinates of the embryo. So what the maternal components do essentially tells the embryo, you're the anterior part, this is the posterior, this is the dorsal, and this is the ventral. And you have the two tips of the embryo which define the ends. So when the embryo is laid in, all the divisions and the gastrulation movements that take place are already aware of what is going to be the anterior part of the embryo and what is the posterior part, what is the dorsal, ventral, and the two terminals, the tips of the, or the two ends of the embryo are predefined. So the genes that contribute to this are called the coordinate genes. And this is set up by the maternal components. So the screens that were done led to this fundamental identification of maternal effect genes. And these are either RNA or proteins or cell surface receptors or ligands that contribute to the setting up of these axes. Now, what was the point? I forgot. Oh, I lost my thought. Yeah, sorry. So what happens? So now you can imagine that the mom is doing all of this. Now, when do these processes get initiated? Because that means as the egg is developing, all of these gene products need to be made by the egg and then laid into it, so that once the egg is fertilized, these coordinates or these different coordinate axes or the different axes of the embryo are specified. So fertilization just gets the thing going. I mean, it just activates the system to be able to now start developing. It doesn't have to worry about, okay, now I have to define the anterior or the posterior or dorsal or ventral. No. So fertilization doesn't do that. It is pre-decided by the mom. It is pre-set up by the mom. And fertilization, the process of fertilization just now gets the ball rolling for development. Okay, so what I meant about the coordinate axes are this. So this is the anterior and the posterior. This is established. The dorsal ventral, so what is the back and the tummy, gets established. And the terminal system, as in the two ends. I mean, you know the tip of the head and the tip of your toe are the two ends. So that's exactly what the terminal system does. Now, you can see that it is written that they mention localized mRNA nanos, localized mRNA bicoid, torso, tall. So what are these? So essentially, what this is telling you is that there is asymmetric localization of the mRNAs, of these certain mRNAs that are mentioned here, in different segments of the embryo. And nanos, like here, nanos mRNA is localized to the posterior system, posterior end. Bicoid is localized to the anterior end. And for the dorsal ventral system, it is the transmembrane toll receptors. Toll is another receptor in immunity, which won the Nobel for innate immunity. Again, a fundamental discovery in Drosophila. But it is very interesting that the development utilizes an immune gene for specifying its dorsal ventral axis. Anyways, that was just a thought that I shared. So the toll receptors are again asymmetrically localized. And then there's these torso receptor. These are all RTK. The torso is an RTK receptor tyrosine kinase receptor, which is again localized at the two ends, the two termini of the embryo, which specify the terminal system. So essentially what this slide is telling you is that there is asymmetric localization of mRNA. And you can then imagine that these mRNAs will then be translated in these specific regions. And now you can see how different the two ends will be. So even though like I mentioned last class, that even though everything is syncytium and the nuclei are dividing, the cytoplasm may not be homogeneous across the two, across the embryo. So these asymmetric mRNA localization will result in cytoplasmic content, which is not going to be the same, and result in, which will be important because now these are all essential transcription factors or signaling molecules. So however the animal reads their concentrations, the development will proceed accordingly. Am I clear?
Yes.
Okay, great. Okay, so
Hello.
yeah,
Hi, I have a question.
who's this?
Nishant from GS 2018.
Okay,
Okay,
hi.
I wanted a question. So at this point, has none of the embryos genome been activated?
At which point?
At the point we are speaking of, like, right?
Oh yeah, no, at this, so I'm just talking about the very early stage of the embryo.
Right.
So
So
now post-fertilization, this is the embryo, but the egg itself, even before fertilization, is going to be like this. So this is way before, this is the egg, and even the early embryo is going to look like this.
Okay. So like I had a question as in, like, so this embryo still needs a decent amount of energy to like sustain itself and all. So do all the energy synthesizing or like, energy production enzymes, like,
Yeah.
Is that all provided by the mother?
Yes. So a lot of nutrients, amino acids, proteins. In fact, a lot of protein is deposited. Now, the point is it required for degrading and generating amino acids?
Or rather ATP or something?
Yeah. So mitochondria, all of this, and in fact, loaded with sugars, is very high in trehalose, stored sugars, amino acids. So it's very energy rich, and the mom has laid it. But what the other thing is that I find very interesting personally is that a lot of protein is deposited. So and the level of ubiquitination of these proteins is very high. One of the questions that I ask myself is that, okay, fine, these mRNAs will be translated into proteins, and also the proteins of these, the deposited mRNAs are anyways present, not bicoid or nanos, what I intend to say, but there's a lot of protein deposition. And one of the things that I feel is that protein undergoes a lot of turnover at this point of time, just based on the ubiquitination status. And one thought that I have is perhaps the protein turnover is also another means to support amino acid supply, because what is happening in the first 24 hours of the embryo is that it's not feeding, right? And the animal doesn't even feed until the first or little later first insta larvae. So as soon as the larvae embryonic development is completed, the larvae doesn't start to feed right away. And while there might be a lot of amino acids and sugars deposited, there's complex carbohydrates and proteins, a lot of glycosylated proteins that are deposited. And maybe they're also, and their turnover is another means to support supply of nutrients. So that's, I mean, that's a purely speculative thought, but that is another way to support the energetics of what the animal might be requiring.
Yeah, thank you. It was really beautiful. Thank you.
Okay, great. So, yeah, now essentially so I talked about these 4 different systems, right? Now, what is common about the 4 different systems is there is, as I mentioned, that there is one asymmetrically localized component, correct? And that asymmetric localization itself results in a spatial coordinate, correct? And this is genetically controlled, meaning there is a gene that controls this particular region. So if you look at anterior, bicoid controls anterior, nanos gene controls posterior, torso will control the terminals, and toll will control the dorsiventral. So each coordinate is controlled by a gene product. Second, it is not symmetrically distributed, as I mentioned. It is asymmetric. So that itself results in some level of kind of spatial information, like the GPS of the embryo. And the other unique thing that you guys have always been, I think, fascinates everybody is the concentration gradients or the gradient formation. Because gradients are a useful way of defining what one can do, what a cell can do, depending on the kind of gradient it senses. It decides its the downstream effect are different. Because if you can imagine the concentration gradient of a transcription factor can result in differential gene expression, because a higher concentration gradient of bicoid can activate genes which have probably poor affinity enhancers, because it's very high, so the poor affinity sites that are promoters can get activated. But the higher affinity ones will not require very high concentration gradients, but the higher affinity sites can be activated, those genes can be activated in even lower amounts of bicoid. So, which will not be the case for the sites for proteins or genes that have very low affinity, very high affinity requirement for bicoid. So you can see that the target genes that are then going to be activated, that see different levels of these transcription factors or proteins, will not be the same. And that kind of sets up a way to discriminate cells. So the ones that have high concentrate, that see high bicoid are high bicoid from the anterior part, and the cells that see low bicoid are more posteriorized. I mean, it's a default system. So higher concentrations of gradient will activate genes that will now specify anterior. But the absence or lower amounts of bicoid will define regions that are more posteriorized. And the presence of nanos does the same thing, confirms posterior identity, whereas these are all anteriorized. Okay, so this is kind of a common component of all the different terminal systems. Now, how does that? So that means a lot of is being done already as the egg is developing. Now we see here actually how an egg develops. This is the Drosophila female germline, and you have these two ovarioles, and this is the oviduct, which is what I was mentioning last class as these. So this is a fully, it's like a flower. I mean, I don't know. It's very interesting if you ever get a chance to dissect the Drosophila ovaries. They're quite, these are individual developing ovarioles. So this is a very mature egg, and these are immature developing eggs. So it has these strings of developing eggs, and each of them is called an ovariole. So they're very, they're like a bud, and you can peel them off and mount them like this. And you can see as you mount each individual ovariole. So this is the tip, and each of this is a developing individual egg. And the more developed the egg is, it's more towards the oviduct. And this mature egg is going to be pushed through the oviduct. And this squishing through the oviduct is what actually activates the egg, pre-activates the egg before fertilization. Okay, so now how does the ovariole look? So this is this is a beautiful image of this entire ovariole. And you can imagine now there are lots of them, so lots of eggs. Essentially, a Drosophila female will lay around 20 eggs, almost 320 eggs per day. So that many, and it lays eggs for at least 5-6 days non-stop. So it's development, yeah. So you can get a lot of progenies from one single fly. Anyways, so at the tip is a germline stem cell, and this germline stem cell is essentially is a multipotent stem cell, and this is a fantastic model in Drosophila to understand stem cell development, maintenance, differentiation, and how does it. So Drosophila germline development is often utilized for understanding stem cell development. So this germline actually undergoes 4 rounds of divisions. Now, the divisions here are not complete, meaning that the cells that form that are connected to each other by these ridges that you can see here. These ridges are called canals, and they connect the cells. Now, after the rise to 16 cells, of which one is going to form the oocyte. And surrounding the germarium is, surrounding the germline stem cell is this cover of germarium, which has these cyst, they're called the cystoblast or follicle stem cells. So they essentially also divide and grow. They form the supporting structure of the germline of the germarium, and they give rise to the follicle cells. Now it is the interplay between, now the one that forms the oocyte is going to be the egg, but the remaining actually form the nurse cells. So it is the job of the nurse cells to be able to, so essentially what the nurse cells are doing are very active in translation, very highly translating cells that are going to be generating all the RNA and the protein and the nutrients that are going to be laid into the oocyte. So all the product that is laid into the oocyte is coming from the nurse cells. So they're going to support the nutrients into the developing oocyte. Now, as you can see here, this is the oocyte, and the nurse cells are highly polyploid, and they need it because they have to be transcriptionally, translationally very high and doing a lot of bulk translation to deposit proteins and also mRNAs that need to go into the oocyte. Okay, so now from just not depositing, the thing that the entire system has to be able to do is also define the anterior, posterior, dorsal, ventral axis. Now this is where things get very interesting, because the nurse cells will deposit what they have to. But now you can imagine whatever the nurse cells are generating, they deposit, the canals that I said that they interconnected actually allow the movement of material into the oocyte. And now whatever the deposit into the oocyte has to be localized spatially at different regions. Because once they get localized within specific regions, then you're beginning to define the coordinates. So we'll see how that happens. Now it first starts with the generation of Gokun. So Gokun is synthesized by the nerve cells. It's a ligand for the torso receptor that I talked about. So this is going to essentially activate the torso signaling pathway. Torpedo, not torso or torpedo. And now what happens is that these nerve cells, they synthesize Gokun and it is transported into the oocyte. Now Gokun actually stays very close. So this is, you can see, I think green here is Gokun. It stays very close to the oocyte nucleus, but gradually, as the oocyte is also growing, it stays along with the oocyte nucleus and activates the Gokun receptor in the terminal follicle cells. Now, this activation of the terminal follicle cells results in the first stage of specification, which is called posteriorization. So it's not the egg that gets specified. The first thing that gets specified is about the follicle cells. So these terminal follicle cells will get specified as posteriorized. Now, once they are posteriorized, meaning in the activation of the torpedo signaling pathway here, this activation will now set up the organization of microtubules in a specific direction. So it is going to, so essentially things need to move from one end to the other or from here to there. If you have to asymmetrically localize things, you have to be able to place them in the embryo. And how do you place things in the egg or in the oocyte is by developing, organizing the microtubules in a way that the movement along the microtubules is going to establish what is going to be in the anterior end or in the posterior end. Now, the activation of Gokun with the terminal follicle cells will result in posteriorization, and this posteriorization will organize the microtubules in a way that so the microtubules are now going to be organized with the plus end towards this side, posterior side, and the minus end towards the anterior side. So now you can see how the establishment of the microtubules will now direct movement in different locations of the developing oocyte. Now, any cargo that is, so you have the plus-ended motor and you have the minus-ended motor. You have the kinesin and dyneins. Are you guys aware of the molecular motors like kinesins and dyneins?
Yes.
Okay, great. So the orientation of these microtubules will employ different motors. And the motors will be then important for localizing the specific mRNAs or the cargos to different ends, the anterior on the posterior, right? So like Oscar will bind, Oscar mRNA or nanos mRNA will bind to kinesin, which is a plus-ended motor, and move here, whereas bicoid will bind to dyneins, which is a minus-ended motor, and will be transported to the anterior end. So this establishes asymmetric distribution of the mRNAs at the two ends. So this will set up the anterior and posterior part of the egg. Right. Now, one thing that I have, this is essentially what okay. So the one, so I mentioned Oscar. So Oscar will bind, Oscar mRNA moves to the posterior end, and Oscar protein, so it is, sorry, it'll be locally translated there, and Oscar protein binds to nanos. So as Oscar moves, as the Oscar protein is present there, it will localize nanos mRNA to the posterior end. So essentially, the initial support provided by the nurse cells is going to be huge in providing the mRNAs that are going to define the anterior and posterior, essentially bicoid, nanos, Oscar, toll, many others, to which I'll come in a minute. Okay, so now so now I spoke about how the anterior and the posterior specification happens. The other thing that gets localized, that gets specified, as I mentioned, is the dorsal and ventral. So how does dorsal and ventral get specified? So the dorsal ventral specification is yet another interesting phenomenon. So what happens is that initially the nucleus is more towards the posterior end, and gradually the nuclei then migrate, and the nuclei has this glucan protein. So the glucan protein is very localized around the nucleus, and so here it activated at this end. It activated the terminal follicle cells and defined one termini, and gradually moves along towards the anterior side, towards one end. And the glucan protein here will now activate the torpedo signaling. Yes. So it'll activate torpedo signaling in this subset of follicular cells, right? Now this, again, very localized activation of the follicular cells, this interaction will now set up the dorsal ventral axis. Now, this is very interesting because the localized activation of torpedo here in these follicle cells will inhibit the expression of genes that are going to be now seen in the ventralmost follicular cells. So the glucan torpedo signaling here will dorsalize follicular cells by repressing expression of genes that are otherwise going to be seen here. So essentially, again, another way to set up a dorsal and a ventral polarity. Okay, so
Hello.
go ahead.
Hi, Tina, you have a question.
Can you just introduce yourself to me, please?
Nishant again.
Okay, great, Nishant. Yeah, tell me.
Yes, so initially you said that the nerve cells produce the gherkin and it goes and it diffuses. And I guess it diffuses and it binds torpedo in the one side of the embryo, resulting in pasteurization.
Correct.
So is this concentration dependent? Because if it's produced on the other end, how does it not bind things on the rest of the embryo and only bind here and result in pasteurization?
So the nucleoplasm around this has restricts. So gherkin is only localized. The gherkin mRNA is only localized around the around the nucleus. And my understanding is that the gherkin mRNA binding proteins are also specifically surrounding. Specifically localized around the nucleus. It is translated locally, and as a result, it activates it activates the torpedo signaling wherever the nucleus is. So the surrounding nucleoplasm is rich in gherkin mRNA binding proteins, and gherkin is then, as a result, very locally translated there as well.
I see. So in the initial image, the nucleus was closer to this end.
Correct. So if you see the developing this thing, you see how it is localized, and then it likes to maintain itself around the nucleus.
Okay.
Right? So again, translation, localized translation, RNA binding proteins generating, localized translation becomes very important for establishing all of this. Okay.
Good. Thank
Is
you.
it clear?
Yes, thank you.
Hi, I had a question.
Mm-hmm.
The gherkin binding to torpedo towards the posterior end leads to the formation of the posterior end, but the same event leads to the formation of the dorsal end. How does the same interaction cause two different things?
So it activates torpedo signaling at both sides,
Yeah.
and at one place it will transport nanos and OSCA. Right? So here the microtubules are going to be key in directing the plus, minus, and the plus-ended cargo. Right?
Okay, yes.
So the cargo that is going to be set up here and here is not going to be, is what is defining anterior and posterior.
Okay.
Correct?
Yeah.
Now, for the dorsal and ventral, dorsal and ventral is for the entire embryo, will be for the entire embryo, right?
Yes.
Correct? So what you will subsequently see is that this initial activation of the torpedo signaling says you're dorsal, and everything else that is not dorsal is going to become ventral.
Okay.
So the activation of this signal doesn't mean it is not going to be, so this is anterior dorsal and this is the posterior end of the dorsal side. Everything here is ventral. This is anterior ventral and this is posterior ventral. Getting my point? But the signaling that will now be set up in the follicular cells here is going to be quite different. You'll see that now.
Okay.
Yeah, so that's what your answer to your question is. So what happens is that the follicular cells are going to see the torpedo signaling, right? Now what the signaling does is
Yes.
Sorry?
No, I was just saying, I thought you were, nothing.
Yeah, I'm going with the class. I think this is, you see this and you will have an answer to your question. Okay, so so the gherkin mRNA is going to be translated locally, and then although although the receptor is present everywhere, it is not going to be activated everywhere. Only at this end, torpedo signaling will be initiated. Now what this does is that it generates a repressor which will inhibit, bottom line, it will inhibit the activation of toll signaling. So it will generate a transcription factor called mirror, and mirror inhibits the expression of pipe. Pipe is so this is all serine proteases which are mentioned here. I mean, so the activation of toll pathway sees a series of serine protease cleavages to generate an active ligand. So what this essentially boils down to, what I'm trying to say is that the localized activation of torpedo will inhibit the generation of the ligand Spätzle that leads to toll pathway activation. So here in these cells, the presence of the inhibitor or the repressor is going to inhibit toll pathway activation. So this becomes the dorsal. So these follicular cells will have the inhibitor, the repressor, and the ventral follicular cells will now are free of that repression because they don't activate torpedo signaling. So they don't see mirror, which is the repressor. So they are able to synthesize pipe, and pipe then actually initiates a series of events. I'll come to that in a bit, which will result in future as the embryo gets fertilized, will result in the ventral specification. So the oocyte actively specifies dorsal fate, but so this is done by the mom itself. So there is a bit of a difference here, because the follicular cells here are repressing ventral fate because they don't have the inhibitor. They have the inhibitor and they actively are dorsal fate. The other end of the follicular cells are not, they're not making the inhibitor, but they have not acquired a ventral fate yet. I mean, they're potent to become ventral, provided they have the activation of the toll pathway, which is seen later on post-fertilization during the formation at the time when blastoderm development is happening. So, so with the steps of fertilization stepping in, the nuclear divisions happening, and the syncytial blastoderm formation, is when the activation, the specification of the ventral cells of the embryo is going to happen. So what is in the oocyte is the specification of the dorsal fate of the follicular cells, that and the lack of this in the ventral follicular cells sets up in a way that the embryo now, the embryo post-fertilization, will develop in a way that the ventral side, ventral follicular cells, will define the ventral cells of the embryo. Do I make sense? Is it making sense, or is it confusing? Okay, I mean, pin drop silence, so I understand it is very confusing. So, so what I'm trying to say here is that this is this is the oocyte. The follicular cells are not the part of the egg, right? They're just the support cells, and they're going to form the supporting vitelline is going to give rise to that vitelline membrane. And the specification of the dorsal cells, unlike the anterior and the posterior, which is in the egg, the anterior and the posterior is happening as the cargo within the egg is being asymmetrically distributed. Now, the dorsal and the ventral fate of the egg is going to be established through the follicular cells, but the cells of the the ventral cells. Now, when fertilization happens, the ventral cells of the embryo have to be told you are the ventral cells, and that is happening through the signaling coming from the ventral follicular cells at the time of blastoderm formation. So what happens is that, maybe if I go over the pathway, it will become more clear. Now, so the ventral follicular cells will synthesize pipe. Now, what pipe does, it sulfates some proteins which are present in the at the ventral in the ventral follicular cells. Now, these sulfated proteins are expressed on the surface. Now, these sulfated proteins will be important in binding a particular protein called gastrulation defective. So they're going to be secreted and they bind to gastrulation defective. And so these sulfated proteins are expressed on the ventral follicular cells, and they will localize gastrulation defective at the ventral follicular cells. Now, gastrulation defective, this is a serine protease, so localization of it here will result in a series of serine protease mediated cleavages. So each of them is these inactivated enzyme kind proenzyme, and once it is bound, it will autocatalytically cleave itself and secrete, bind to another proenzyme, snake. Binding to this autocatalytically activates snake, which will cleave itself, and like this, it will result in a series of the cleavage, ultimately resulting in the cleavage of Spätzle, which is the ligand for toll. Spätzle will bind to the toll receptor. Now here you see that the cells of the embryo are going to be seeing active toll signaling. So from the follicular cells to the perivitelline space, so all of this is very in the fluidic space of the vitelline in between the embryo and the vitelline membrane. So these are secreted events, finally leading to the activation of Spätzle, which is normally present in a pre-form. So pre-Spätzle cannot bind to toll unless it's activated by cleavage. So this whole process of gastrulation defective all the way to generation of active ester, which will then cleave Spätzle, result in the active ligand that will now toll is present in the entire embryo. All cells express toll, but all cells do not activate the toll signaling pathway, precisely because the synthesis of pipe is going to be very localized in the few ventral follicular cells. Now, this is going to restrict toll activation in a subset of cells of the embryo. And so once toll is activated, so dorsal is your NF-kappa B, the most common NF-kappa B. So normally dorsal is, it's a transcription factor, and it is bound and kept out of the nucleus. So cactus is a binding partner for dorsal, which prevents its entry into the nucleus. Now, once toll signaling is active, it will result in degradation of the of cactus, and dorsal will now be free to go into the nucleus and activate the transcription of genes, which are now going to be defining the ventral fate of the egg, of the embryo. Is that clear? Does that make sense?
Yes.
Okay.
Yes.
Okay, great. So from the ventral follicular cells all the way to now defining the cells of the egg, this is how the signaling goes. So you have membrane, vitelline space, and then another membrane component of your embryo. Okay, so now how does it look? So as you can see here, so all essentially, if you do a toll staining, receptor staining, you will see it everywhere in all cells of the embryo. But like I just mentioned, you're seeing active dorsal. So even if you do dorsal staining, you might see it everywhere. You will see it everywhere. But it is the nuclear localization of dorsal which is not seen everywhere. The nuclear localization of dorsal is only seen in these few cells, in these specific cells, the ventralmost cells. Now you can appreciate that the expression of dorsal is also not uniform across all these cells, the highest being here, and gradually it reduces. So the protein expression also has a bit of a gradient here, which means that the distribution of dorsal all along the ventral region is not uniform. And this sets up the gradient of dorsal. Now what this gradient of dorsal will do, the reason why am I stressing on dorsal is because this is where the first morphogenetic movement of gastrulation is going to initiate and divide the undifferentiated cells. I mean, there's no segregation of the three germ layers. So it is the distinct presence of dorsal and the subsequent movement of the inward movement of these cells, which is now going to divide and give rise to the three germ layers. So the mesoderm, ectoderm, and the amniocerosa. Sorry. So now the highest amount of dorsal seen will define the mesodermal cells. The gradually lesser amounts that are seen will define the ectodermal cells. And the ones that do not see dorsal at all will be actually the amniocerosa cells. So now, in the absence of dorsal, so how did the name dorsal work? So Drosophila biologists have this habit of naming genes after the phenotype that is seen. So it's very confusing. So essentially in a dorsal mutant, the embryo is completely dorsalized. So which is why when it was identified, when the gene was identified, the embryo was completely dorsalized because there was no ventral cells. So hence the mutation in that the gene got named dorsal. But essentially it is the lack of ventral formation in dorsal mutants. So the presence of dorsal specifies ventral fate. So there's this like the opposite. So if it's dorsal, it's not that the dorsal phenotype is missing. In fact, the whole embryo is dorsalized. So which is why right here that dorsal inhibits dorsalizing genes. So the presence of absence of dorsal in the amnioserosa in these topmost cells, which will give rise to the amnioserosa cells. Okay, I think these are 3 different mutants. I'm forgetting the 3 different mutants. So first one is wild type. Right. So this is loss of dorsal. So everything is dorsalized. And this is gain of function of toll signaling everywhere. So essentially dorsal is in all cells of the embryo. So this is a completely ventralized embryo. So this is a completely dorsalized embryo where there is no toll signaling, no dorsal into the nucleus. And this is a completely ventralized embryo because dorsal signaling is seen everywhere. So this is how the mutation in dorsal or gain of function or loss of function or gain of function of dorsal activity is going to affect the specification of the dorsal ventral axis. Now this is probably the first example of a gradient that I mentioned here. We'll come to picoid next class. So the effects of dorsal mediated patterning is, now this is a transcription factor, NF-kappa B, in mammalians is what is commonly known as. And dorsal will result in active transcription of its target genes, twist and snail, and rhomboid. Now, like I mentioned, there are these high affinity, low affinity sites that are going to be very critical here for sensing dorsal concentrations. Now, since snail, twist, and rhomboid have the highest affinity, have the highest affinity enhancers. Snail, twist, and rhomboid actually have the lowest affinity enhancers. So, which is why they're going to be activated only in cells which have the highest concentrations. But subsequent to these cells, the genes which even have very high affinity enhancers are going to be seen here because dorsal concentration is less, but since those genes have high affinity enhancers, they're going to be coming up in cells with low dorsal concentrations. Now, so twist, snail, and rhomboid are going to be seen in all these cells. Now, this the other thing here that I want to mention, twist is a very commonly, twist and snail are very commonly studied for EMT, is epithelial to mesenchymal transition. This is essentially what is happening here as well. So the same transcription factors that are very important for EMT and going to make cells more migratory is the same module that is going to be, is seen here and is under the influence of the toll signaling. So the activation of snail is going to, so snail is going to be repressing ectodermal fate, whereas the activation of twist is going to be activating mesodermal tissue development. So snail will be actively repressing ectodermal genes. So which is why these cells will become mesodermal tissues, give rise to mesodermal tissues. And the cells which are devoid of twist, devoid of snail, will be now becoming neuroectodermal cells. Now, so now the important part here that is initiated is the inward movement of these dorsal cells to give rise to the ventral furrow, and that is initiated by FGF signaling. So at this end, the green end, where so FGF receptor is present in the dorsal cells, but the ligand is not seen everywhere, only in places where the snail component has been, so cells which are not expressing snail, so it's not repressed, FGF ligand expression is initiated. And these cells are adjoining the mesoderm and the ectoderm. So these are called the mesectoderm cells. Now these, at the interface of the mesoderm and the ectoderm, these cells will make FGF ligand, which will activate FGF signaling in the dorsal cells. And this will result in the inward movement of these cells to give rise to the ventral furrow. And that is the first movement in gastrulation that is now initiated. Afterwards, then everything else is set up by the movement that this one initiates. The ventral furrow initiates the subsequent steps of movements. Is everyone with me on the same page?
Yes.
Yes.
Yeah, not too complex, right? Okay, so okay, so now what essentially we have okay, I'll stop here, but what I want to recap here is that I hope we've been able to understand or appreciate the contribution of maternal components and roughly how maternal components or mRNA deposited in early developing oocytes or proteins deposited in early developing oocytes can result in asymmetries and specification of different cell populations. So, what are the means by which, if I have to generate asymmetry and I have to generate axis, if an embryo has to generate axis and asymmetry, roughly what are the mechanisms that could be employed? One is localized translation, as for is seen for gherkin mRNA. Then secondly, the utilization of microtubules and the different motors, the plus and the minus ended motors. So depending on the kind of and depending on the cargo the motors bind, some cargoes bind to plus ended motors, some cargo bind to minus ended motors. So depending on how the system establishes the microtubular polarity, you can segregate or separately maintain RNA and proteins at distinct locations in the oocyte. So that's another way to generate asymmetry. And as they are separated or as they are maintained, the other way that asymmetry can be generated is by the utilization of the concept of gradients. Because gradients of transcription factors or gradients of ligands. So gradient of transcription factor here, I refer to dorsal. Gradient of signaling, I refer to the presence of pipe and subsequent activation of Spätzle, and how it diffuses in the vitelline membrane to give rise to active toll signaling will result in gradients of toll activation. So localized mRNA translation, utilization of microtubules, and gradients, these are essentially the three fundamental ways by which the oocyte and the early developing egg is going to be generating asymmetry. So are these points clear to everyone?
Yes.
Yes.
Okay, great. Now I will leave you, I'll get back to the question that I asked in the first class. Not a question, but a general discussion. But I will leave you guys with this particular slide. Now, this slide essentially is talking about this that I mentioned. You have a maternal component and you have an embryonic component. So, how was this pathway actually even discovered is what that experiment talks about. And I would like you guys to look into this slide and understand this, and maybe we can talk about it next class. It's a very simple experiment, but I want to see if you understand what was done. So everybody has the access to slides, right?
Yes.
Okay, so this particular slide that I have here, and I don't know if you have been circulated with this paper, but this is another paper that I want you to read. Essentially what I talked about is years of work put in by Lustig Folhard lab to establish this dorsal ventral polarity. And you can imagine it's not a simple signaling pathway. And it's not that it's happening in the same cells in the same location. And now we have a lot of technology at our disposal to be able to elucidate anything. But back in the 90s, when just transplantation or just some basic molecular tools were available, they could actually establish this entire pathway. However, they did it. So that I want you guys to read that manuscript, which is here, and how they identified these extracellular activities using genetics and some bit of transplantation. They could delineate the entire pathway that establishes this dorsal ventral polarity. So this is, if we are able to do a discussion on the paper Friday, that will be great, because this is a very nice paper. I really like it. It may be a little complex for everyone to read, but I think it's a very fascinating manuscript, because just the way they did those experiments and how they identified the dorsal ventral polarity is very fundamental. So bringing down to the idea that you don't need complex set of experiments to prove a point. It is the very most basic and simple experiments that can prove a valuable point. So I think that's the reason I'm emphasizing on this manuscript. Okay, so, but regardless of that manuscript, this is one simple thing which is here in the picture that was done by Trudy Schubach's lab, where they were actually working on the torpedo signaling and identified how the interaction between the oocyte and the follicle cells give rise to this dorsal ventral polarity. Okay, so okay, the last 5 minutes, yes. So what are the thoughts on Nishant, right? Or who was it? You asked the question, right?
I asked, yes.
Yes, do you want to start? What are your thoughts?
And this
So
on
can
the
you just
slide.
can you just not this one, but what I mean about is the question that I posed, a thought that I posed last class, that why what would be the factors, what would be the factors that you think lead to a syncytium-based division in Drosophila vis-a-vis the entire cellular divisions that happen in vertebrate embryos or other embryos.
Okay.
Yeah.
Truthfully, sorry, Tina, I wasn't there last class. I'll think about it. Can I answer in a bit?
Oh, okay. But who was it who initiated that?
It was me, Atal.
Atal, sorry, Atal, I'm so sorry. I'm very sorry, man. Yeah, so go ahead. Yeah, you had, sorry, Nisha, I caught you off the hook. So, yeah, Atal, what's your thought on that, man?
Maybe I'm not sure, but if you compare the Xenopus development with that of Drosophila, I think the Xenopus development is more like it forms a ball of cells which is majorly not that organized if you compare to that of Drosophila, where cells have to be aligned more specifically to form the cellular blastoderm stage. So I think the multiple nuclei and then their alignment becomes more easier if there is no cell membrane already laid around the cells. So I think that alignment is much easier in case of Drosophila, which is not the case in Xenopus, where it is simply cells dividing, cytokinesis happening, and then that ball of cells being formed.
So in one line, can you just summarize? I mean, I'm a little.
You're
trying to say that in Xenopus the divisions are cellular because they're aligned.
No. In Drosophila, they are more. The organization is better if you compare the Xenopus development with that of Drosophila. The cellular blastoderm stage appears to be more organized than the Xenopus development, which is.
Okay, fine. So, but my question is that why couldn't, so cellular blastoderm is much later, right?
Yeah.
So in Xenopus or a chick or mouse, right from the first division, it's a complete cellular division, right?
Yeah.
Karyokinesis with cytokinesis, right?
Yeah.
So the nuclei segregate and the cell membrane separate cells, right? So you have two distinct cells forming right from the first division.
Yeah.
But in Drosophila, the nuclei divide. They align themselves much later stage, 10, 11, they start aligning themselves to the periphery. And prior to that, they're all, they're a syncytium. And it is only once they align to the periphery is when the cellular membrane starts pinching in.
Yes.
Correct?
Yes. So I was thinking because in Xenopus, no such alignment along the periphery is required, so it can simply divide and form a spherical ball of cells. Whereas in Drosophila, since you need this alignment, it will be easier to form multiple nuclei and then sort of move them and align towards the periphery, and then the cell membrane can be pinched off, and that organization becomes easier, is what I thought. I'm not sure.
No. I mean, this is a thought, right? There's no way to say it's right or wrong. Yeah, yeah, okay, makes
Hi,
sense.
hi Tina. I was
Okay.
I
No,
was
go ahead
thinking.
then.
Okay, thanks. I was thinking that maybe the nucleus.
Who's this, please?
Oh, this is Anjum. I am a part of the in PhD incoming in PhD batch GS 2020.
Okay, Anju, hi, nice to meet you.
Nice to meet you too. So I was thinking maybe the nucleus actually waits for the gradient to form, so that ultimately when the membrane is formed, it encloses specific parts of the cytoplasm that already has the determinants then that will kind of help it to differentiate along a specific um, direction. Maybe I don't know. I was thinking.
So go ahead, Tanvi. So you're saying that
Basically, it is waiting for the gradients to form in the cytoplasm of the cell, and then ultimately, when the membrane is being laid out, each of the nucleus is enclosing a specific part of the cytoplasm that has determinants that will allow it to differentiate along a specific direction. I don't know. I was thinking like
yeah,
maybe
yeah,
that.
yeah, 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 makes sense. Yeah, yeah. So kind of facilitating the gradients to
Yes,
be interpreted before and cell identities to be laid out. I mean,
yes,
not cells will form later, but at least the identity of the nucleus is not going to be same. Uh, uh, Whatever genes they transcribe thereafter will be different. So following that, the nuclear identity will be distinct from subsequent segments.
yes. That's what I was thinking.
But um, but again, if I like Atul mentioned that the movement has to happen. Um, Maybe the alignment of these it will be now since there is so again Atul's point was that since in Xenopus you don't need any movement of such a sort, which will be better with Drosophila syncytium, right? That's your basic point, right, Atul?
Yes.
Yeah, and your point is uh, Anjum, your point is about mentioning, I mean, you're mentioning essentially that the gradients will be facilitated better
Yes,
and the identities will be created better. I mean, it'll be established.
yeah.
But if I argue these concepts will be common to development of all other organisms, right? Movement, gradients. So essentially, what both of you are trying to say, it buys in time. It facilitates. They will be done. They will be done faster, perhaps,
Yeah, that
because
is also what I was wondering, because if there are more nuclei in the same cytoplasm, then the speed of development is also increased, which is what we discussed in the last class also. So I kind of compared how long it takes for Xenopus to sort of reach the stage before it gastrulates, and similar to I also compared that to Drosophila's time. So they weren't very different, but yeah, Xenopus takes, I think, more time to start gastrulation in comparison to Drosophila. So I think that might be one more point.
correct. So I mean, I personally feel now like what you guys are all telling me. I feel that you're on the same page as I am. That this syncytium is one way to speed up development. So having to cellularize every time it divided would have really elongated the timings of the early embryonic development. Um, I am totally in agreement with that thought. I feel cellularization would have taken a lot of time. So let's get 256 nuclei lined up, and then once there are 3,000 nuclei, let's do cellularization now one time cellularization is done, because the early, and essentially the fly is not going to live like the humans or the frog or the fish or the, I mean, it has a very limited lifespan, so its embryonic development is also scaled accordingly. It can't spend days in embryo. So I think what you guys are making the point that it's faster. Yes, I think so too. I had one point, I don't know if it makes sense, but the other point I thought is the nuclear to cytoplasmic ratio. That is really low, because the chromatin amount is much less compared to the huge cytoplasm that it holds. Maybe that is one reason that inhibits cellular divisions. I mean, so the point is what keeps cellularization off, right? That was the other thought that I had, that what would keep cellularization off. And we know that nuclear to cytoplasmic ratio is very important for cell division to happen. And if you look at the Drosophila embryo, the nucleus is one tiny component in the beginning, and the cytoplasm is huge. Now, if that is true, if that is true, and I think the experiment that I mentioned last class about Bruce and Edgar, where they halved the nuclear content, so the nuclear divisions were delayed, and the cellularization was delayed, development was delayed. So which means that the embryo is evaluating nuclear to cytoplasmic ratio every time. And as it attains a certain nuclear to cytoplasmic ratio is when cellular divisions kick in. If this nuclear to cytoplasmic ratio is altered, then cellularization can be sped up or delayed. And that was the other thought that I had, that this might be one reason why the cellularization is also not happening right from the beginning. Because if you see in the beginning the images of here. So from stage 1 to 2, even if the nuclei is divided, how would it even it's asymmetrically placed? How would even the cell membrane divide all of them? So it's just like tiny nuclei floating in an ocean of yolk. So this might be another reason that cellularization is kept off until when the nuclear to cytoplasmic ratio in the cells have been established. So based on that, I think I'm, because of the Edgar's experiments, because of some experiments that have been done in these directions, I feel that may be another possibility. Yes, definitely time and speeding up, because 8 minutes division will not be possible with cell pinching in. But there has to be an active mechanism that keeps cells from pinching in at this point of time, say, off, right? So even when the cells the nuclei are aligned, the cell divisions could have initiated here. They don't. They only do after a certain number of nuclei have been achieved. So even after alignment, if the cells could have been separated and then the divisions could have continued, but not just to maintain a certain rate, but I also feel the embryo is constantly evaluating the nuclear, the chromatin to cytoplasmic ratio. Yeah, that was my point. Any other points that you might have? Anybody else?
Hi, Tina. This is Utkarsh here. So,
Utkarsh, okay. Hi.
hi, Tina. So I was thinking, like, can this be explained in any way this could be because of metabolism in some sense, as in that is it that the embryo could be in sort of a metabolic state which is sort of restrictive for cell membrane synthesis, and sort of doesn't allow that, in the sense that because it's again, there are like a lot of costs that would be involved in synthesizing the cell membranes.
Correct. I mean, absolutely. I totally. So division. So that will be the time-consuming event that you have to generate new lipids to make membranes, and already you are you have a lot of energy that needs to be generated to divide the nuclei. And the tremendous actin dynamics that is going on here, which will require a lot of energy. So there is definitely a fight for energy, but absolutely. So energetics will be key factor in deciding whether I want lipids to generate membranes, or if I have lipids that I need to break down to generate ATP to drive the actin dynamics that will be driving my cell nuclear divisions. Absolutely. So energetics is going to be a deciding factor. But to me, I mean, even to me, I find these early. So maybe at this point of time here, when the when the embryo has a lot of nuclei, what keeps the cells from still pinching off? I mean, here the nuclei are floating and there's a lot of yolk sac. So I would assume that if the embryo had to pinch off into two, could have done, right? Unless it but there's going to be limited resources, as you're mentioning, and it is already cognizant of the fact that this is a non-feeding embryo, so it's not going, and the nutrients are always going to be limited, and that is going to restrict its cellular divisions. Yeah, that's possible. That's another thought. Yeah, that's very nice.
Hi, I'm
Hi.
Meghna.
Meghna.
Yes.
Yes.
So I was wondering, could it be because of the overwhelming influence of the maternal components? Because maternal components influence the development till such a late stage of development that it's easier that all the nuclei are in the same cytoplasm in which the maternal components were initially laid.
Yeah, I think you're also referring to the same point that Anjum mentioned, right? So the establishment of gradients or things will be much more faster facilitated if they are in a syncytium, right? That's what you're trying to say, right?
Essentially, yes, because I mean, if the cytoplasm is already divided, maybe the maternal components will not be available in the entire area. It would be if all the nuclei were in the same cytoplasm. The initial localization would be better maintained.
Yeah. I mean, yeah. So I think the regulation between different nuclei will be easier in a syncytium. Vis-a-vis, if there are two independent cells, then it'll be more time consuming. You'll have to have more different means of coordinating two cells to behave one way as opposed to two nuclei with a connect. Although the cytoplasm is not really connected, not really diffusing, so they're quite compact, arranged around the nuclei. So it's not as freely. Yes, gradients can be easily established. That is definitely true. So there is luxury of certain molecules to diffuse much better as opposed to other molecules which are very tightly arranged around each nuclei. If you see images, you will see that some nuclei are some proteins are very tightly very compact around the nucleoplasm, and some like bicoid mRNAs can diffuse. So yes, I think what you're saying is probably another way of facilitating faster divisions.
Yeah.
Yeah. Maybe something to read upon will be, so I think what if there is an active control on, so the point of cellularization initiates at the stage 12 and 13 when actin and microtubule dynamics becomes very critical um, in generating the individual cells in cellularization. So somewhere actin control on actin polymerization probably inhibits it from happening at earlier stages. So actin dynamics may be another factor that pitches in. So I don't know how much actin dynamics is possible at earlier stages. Because if we were to divide the whole embryo into two, it will require a huge amount of actin control. So whereas actin is available for the nuclear divisions, the chromatin divisions, the actin that now becomes available for generating each individual cells is possible only after a certain stage. So that may be another way to look into it, that how is actin being regulated early and late when cellularization happens. So that might be that may be a mechanism that keeps it off from happening earlier. Because once you have a good number of cells, the cell membrane could have divided, but it didn't. What would have kept it from not dividing? So maybe earlier stages, the syncytium and the gradients have formed. But after the gradients have formed, why did they not divide? Why did they not cellularize here? Why did they choose only later on? So another thing could be the actin dynamics, maybe, or the proteins that are involved in cell divisions become only available after zygotic genome activation happens, is when they are more, because cellularization is seen around the same time when the maternal to zygotic genome activation is also happening. So both of them are just around the same time. So I don't know, but that may be one thought I had. Okay, so any more? That was good. That was very nice. I'm glad that people thought about it. So any more points anybody has? Okay, great. So I'm taking that silence as no. And I'll stop here for today, and we will resume Friday. And Friday we will touch upon anterior, posterior, and following this, how the segmentation happens and the entire body plan that gets laid, and how do the two genes, bicoid and nanos, start doing this. Okay, so, but food for thought is read this paper. I don't know if you will be done by Friday, but definitely Monday will be one day we can take it up and see how the class has found this paper. Good, bad, great, terrible, don't like it, would never read it. Those thoughts, any thoughts will be appreciated. But Friday, we will go over this experiment and see what this experiment means. We'll start from here and go to the anterior posterior axis determination. Okay, sounds good?
Yes.
Okay, great. Thank you guys. I'll see you Friday.